Assalamualaikum and hi to my lecturers and my fellow friends. My name is Nurul BINTI AZAN S55824. So today I will present to you my research proposal presentation. The title is Serenity Impact on Growth, Oxygen Consumption and Expression Pattern of Selected Candidate Genes in microbe Scylla tuberca. So this is my table of content. For the introduction, I've been choosing Scylla tuberca as the one of the microbe species. As you can see from the taxonomy, it came from phylum Arthropoda, class Malacostraca, order Decapoda, family Protodontia, and the genus is Scylla. For the distribution, it can be found in the Eastern Indian Ocean, Northwest Pacific, Western Central Pacific, Indo-West Pacific. This microbe can be found in the coastal seas, estuary, coastal lagoon, and mangrove. Under stressful conditions, crustaceans deploy a set of candidate genes for rapid acclimation to survive. More energy needed to minimize the impact result in reducing the growth and reproductive success of crustaceans. For animal at this statement, crabs and other crustaceans are not considered as animals. For the justification, salinity can be increased with human activities. Secondly, microbe is commercial farming and high demand in Southeast Asian countries. Scylla tuberca is not the common species that undergoes any experiment compared to other species before this. There are two objectives from this research, which is firstly to investigate the response of microbe towards different level of salinity. Secondly is to examine the changes in oxygen consumption rate with the expression pattern of osmoregulatory genes. For the literature review that's studied by Matlifat Rahul et al. In 2020, for the finding is firstly environmental salinity will affect severe osmotic stress that resulted in growth and rapid changes in expression pattern of candidate genes. Secondly, salinity could influence the distribution, abundance, general physiology, survivability and well-being of crustaceans. Third, the major challenge for crustaceans with salinity changes is to deal with ionic balance between body fluid and surrounding aquatic environment. For the knowledge gap, further study can use more genes and other biological aspects. Firstly, biochemical changes such as changes the amount of fatty and amino acid. Secondly, cellular changes such as gill structure. And third, physiological changes such as metabolism rate. There are three research questions, which is firstly, how salinity will affect the Scylla tuberca farming in the future. Secondly, what is the relationship between salinity and the oxygen consumption rates? And third, what is the correlation between growth, oxygen consumption, and expression of selected genes? For the methodology part, there are 4 flow methods in this research. I will go one by one. The highlighted here is 30 juvenile Scylla tuberca will be collected from a local aquaculture farm. First method is experimental tank preparation, sample collection and maintenance. Firstly, 3 experimental tank, which is 3 different salinity with 10 replicate. Second, 30 collected juvenile crab will re-array for 10 days. Next, the control will maintain in 20 ppt with continuous aeration. Fourth, each tank been partitioned with the plastic sheet create 10 individual chamber. Fifth, salinity will reduce 2 ppt per day, which is reduce 1 ppt and wait about 4 hours and reduce another 1 ppt. 7. The uneaten food materials will siphoned out daily to maintain the water quality. Second method is evaluation of growth performance. Firstly, crablets will maintain in the salinity for upcoming 3 months to examine the growth performance. Secondly, before and after the total body weight of the microbe be measured. Third, 9 random crab, which is 3 individual from different each salinity, to wait on a digital balance. The third method is measurement of oxygen consumption. Firstly, 250 ml flow respiratory chamber were used. Next, 4 hours were taken to measure the oxygen consumption rate of microbe. Next, oxygen was deducted during oxygen enter and oxygen exit from each chamber. Fourth, maintain the rate water flow for 1.5 liter per hour. Fifth, salinity induced changes being calculated using formula. Next, oxygen consumption experiment being adjusted with the control chamber with no crabs. Next, 3 replicate individuals from each serenity level were used to measure the oxygen consumption. And last method is gene expression study. First, gill and hepatopancreas tissue of crab were collected and preserved in buffer. Next, 5 crabs were examined from each serenity for 5 biological replicate. Next, the preserved sample were kept in room temperature for 12 hours and maintained negative 80 degrees Celsius RNA integrity, and after that it thawed on ice. Next, RNA extracted using RNA isolation kit followed the protocol using DNA enzyme. Next, purified total RNA sample were converted to mRNA and cDNA using cDNA synthesis kit. Next, cDNA sample were preserved at negative 20 degrees Celsius for subsequent analysis involving real-time quantitative polymerase chain reaction, which is RT-qPCR. Next, RT-qPCR will perform in 20 microliter reaction mixture and placed in the real-time cycler. Next, to ensure the amplification of a single qPCR product, a standard Mach curve analysis was performed following RT-qPCR. Lastly, the control represent expression values of 18S. It been calculated using this formula, which is CT represent to gene expression value. Next, for the expected outcome, for the first graph, it showed that all crabs can grow by increasing the mean body weight, but clearly seen that crabs in 20 ppt is the highest among all. Second graph show the crabs in 20 ppt remain the highest among all serenity. Besides, gene expression pattern for crab in 0 and 10 ppt decrease until day 2 and keep increasing after that. Next, R2 values indicate that strong positive correlation were observed between growth performance, which is body weight, and expression of growth regulatory genes, as you can see in figure 9a, and also between rates of oxygen consumption and selected osmoregulatory genes in figure 9b. For the analysis part, it is including qualitative and quantitative data. The main variable are serenity, oxygen consumption, and growth development. There are 2 software being using, which is SPSS and Microsoft Excel to store the data. A few tests were running out, which are 1-way ANOVA, 2-way ANOVA, Duncan multiple range test, and post-hoc test. Proceed to my Gantt chart. Last but not least, this is some of the references that I'll be using for research proposal. Thank you.